I'm going to record this. So, right, so here we are. Let's see. Let me draw a line that's black and that wide. How about that? Okay, so here is, oh look, it's cadence. Oh, I think I just let cadence in. Hold on. Yeah. Okay, so hi Cadence. What we're doing is we're talking about the next step from messenger RNA, and so let's see. I'll do the squiggle line, and here's all of your messenger RNA bases. I'm kind of being a little sloppy here because I want to go quick, but okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And these you know what, I better type. Something like that. Okay, so here's your, I wonder if I can make that bolder. No, I cannot. All right, fine. So there you got your messenger RNA there, and oh nope, stop. I'm gonna draw. Okay, so here's the thing. So this goes into the ribosome, which is, I'm just going to draw it. It's sort of this thing. It's kind of shaped like a hamburger. And as the RNA feeds into this thing, the message gets read, and it so I'll write that up here. So as mRNA feeds through, the code gets read 3 letters at a time, starting at AUG. So there's AUG is called your start codon. I don't know why. What a horrible color, that brown. That's nasty. Every 3-letter group called a codon. Every 3-letter group stands for 1 amino acid. Okay, and if you don't remember, hold on. Hopefully you're still paying attention. If you don't remember, amino acids are what proteins are made of. Let me just remind you of that. That's what's coming out of here is a protein made of amino acids. Hold on. Right, there's a single amino acid, but just to remind you, hold on. Oops, amino acids make proteins, right? And a polypeptide chain is a protein, and it's made of individual amino acids. Yeah, and so that's what the ribosome is making. Okay, good. Well, I just saw your message, Cadence. I'm glad you're here. That's what's important. Okay. So this is getting made in the body. This is getting made all the time in the body. This is happening about 1 billion times in different places in your body right now, if not billions or trillions of times in your body. And And so let's go back to the whiteboard for just a second. Yeah. So we're feeding this messenger RNA through this thing, the ribosome. Oops. Ribosome makes proteins. And as it feeds through, it's going to get read 3 letters at a time. I can't highlight, but I can... What's that? What's that? What's that? Oh, yeah. Well, anyway, let's see. I think... What? Oh, boy. So, yeah, so there's... Nope. Nope, that's not going to work. Basically, it gets read 3 letters at a time. And it always starts at AUG because AUG is start. And so there could be 100 on the other side. Like all, oops, dang it. All of these here, they don't matter. When AUG comes, that's when it starts reading. And then it reads it according to a sort of decoder that I'm going to show you how to use now. And then we're going to practice it. Okay, so let me stop. I'm going to pause here for a second. I don't know if I can bring this. Can I bring a picture into this? No, that makes me sad. That's too bad. All right. Okay, so we'll have to go back and forth. All right, so the thing that I want to show you, you have a copy of this on Classroom. I'll show you where it is. If you go to Classroom RNA Translation Decoder, looks like this. And I know this looks super confusing for a second, but after you learn how to use it, it's really not. Sort of stare at it for a second, but then what I really want you to notice is on the left it says first letter, on the top it says second letter, and on the right it says third letter. That is for reading the 3 letters of your RNA code, and at the end it's going to tell you what amino acid that stands for. This is the process of translation, translating RNA into amino acids, which here's a list of all the amino acids and their abbreviations. So you can see each of these boxes shows you that every single one of the possible 3-letter combinations between C, A, U, and G stands for either start, stop, or one of the 20 amino acids. And the way you read it is first you use the first letter to narrow it down to a row, because each of these, U, C, A, and G, stands for, basically narrows it down to a whole row. So if we're looking at, let's go back to the start, let's go back to the whiteboard for a second to see what our first 3 letters is. Okay, AUG. AUG actually stands for an amino acid too. It doesn't just tell the ribosome where to start, but it also stands for one. So let's take AUG over to the decoder. I'll just use this one here. I have one right here. And I'll open it up in this tab. All right, and so wait, hold on, I'm not sure you can see this. All right, so here's the thing. And so first letter, second letter, third letter. So A is our first letter, right? AUG is what we've got. And so our first letter is A. And what that does is it narrows it down to this somewhere in this row. And what you can do is you could look at this until you see AUG. Here it is right here. And that tells you it's met which is methionine. But the systematic way you use this is that the first letter narrows it down to a row. The second letter, which is U in our case, narrows it down to a single box. And then the third letter part narrows it down to one of these, so within that box. Or you can just look for it. AUG stands for met, which is methionine. And so back to our first whiteboard here. So that means that this first amino acid that gets put on here is methionine. Now it's up here because it got put on first and other ones got built under it. And so, okay, so this second one is CCG. Whoops. So let me put that in the chat. CCG is the amino acid. So if people could please privately message me the amino acid that stands for, so that we can start getting a handle on this. So CCG. So we're going to go over here. Oops. Go back to the thing. All right, CCG. And again, first letter narrows it down to this row. Second letter narrows it down to this box. And in some cases, all four of them in the box are going to stand for the same one. They don't always. And so one, two, three, four, five, six, seven, eight. That's 8 out of 14. Okay, folks, are the other 5 of you there? I want everybody to try, please. So again, what you're doing is you're reading the code CCG. First letter, second letter, third letter. First letter you read from the left, narrows it down to a row. Second letter you read from above, and wherever it intersects with the row from the first letter, narrows it down to a box. And then if you need it, the third letter can tell you which one of the 4 in this box it is. In this case, they all stand for pro, P-R-O, which, if you look, is the amino acid proline. And so yeah, that's fine. It's people make mistakes on this, but once you get it, you get it. So all right, so I would go back to our whiteboard then, and just point out that the second one, which would be right here, oops. Oops. And so then the third one, oops, AAC. Let's do that one together, and then what? Well, we'll do the last 2 together. So, so AAC. Everybody this time, please. Hopefully AAC isn't stopped, and I'll go back to the thing. Oh, is somebody talking? Okay, let's see. 1, 2, 3, 4, 5, 6, 7. Okay, first of all, 7 out of 14. Yep. Okay. Okay, so again, just to what was it? Now I have to look at the chat again to remind myself what it was. AAC, right? AAC. So yeah, letter A means that it's in this row. Letter A again means it's in this column of this row, so that narrows it down to this box. And then AAC is right there, and it's ASN, which stands for asparagine. Okay, all right. And then the last one on our little example here is CCA. So that'll be the last one, and then we'll get on with our lives. So how about let's do CCA. Ooh, did we have that one before? Oh no, we had CCG before. Okay, we had CCG before, we have CCA this time. Okay, that's 4. I'm waiting until everybody does this. I did ask everybody to do this. Okay. Okay. Well, it's proline again, because again it's C C A. Right there it is, right there. Stands for proline, and that's how it goes. All right. So what we're gonna do. Okay, hold on a second. Oop. People talking to me in the chat. Hold on. I knew what you meant. I knew you didn't mean goo. Okay, so let's start the share here. All right, I am going to add a little something for you to work on right now. And it's called Transcription and Translation Practice. I'll show it to you in just a second. Let me put it up on the Google Classroom for you here. Okay. Oh, dang it. Okay, that's dumb instructions. Please complete this worksheet. Oh. Okay, and I got this ready last night. Should be right here. Boy, everything's super slow today. Uh-oh, here we go. No. Okay, we'll make this a 10-point assignment, and we'll put it in week 16. Okay. All right, so that's been assigned. Let's take a look at this real quick. Okay, so transcription translation worksheet. tell you what, let's, oops. I thought I deleted this last night. I meant to delete this, so don't worry about that last problem. And I know there's no number 4. I don't know what happened. Anyway, so what do we got here, folks? So this is a transcription and translation worksheet, and you will use the decoder sheet that I gave you, the translation sheet, and this to figure out what goes here. Now, let me just work through some bits of this example. First of all, this is one of those where you're going to have to adjust this as you go because, well, we'll see what happens. I don't know. It might displace things as you type. But what this is asking you is, like, let's use this number 2 as the example. Number 2 is the most straightforward. It gives you a strand of DNA, and it asks you to transcribe it into RNA and then translate it into amino acids. And so, for example, TAC, what would TAC be in RNA? If I was going to transcribe TAC, so what we did yesterday, basically going from DNA to RNA, AUG. Oh, look at that. Yeah, AUG. That's right. And okay, so there's AUG, and then underneath that, it's asking us what amino acid would that stand for. And if we go to the transcription sheet here, AUG, we sort of already know because we've done this, but A, U, and G is methionine, met. It's also the start. And so you would put that here, M-E-T. Yeah, and that's how this works. So you would do that all the way across. Now with this one, this would be a little bit different. You're gonna have to sort of reverse transcribe this one. Now, luckily, I will say you could kind of cheat and just look at this one since it's also AUG. All of these start with the same one. So I'll tell you what. Let's do this one instead. ACU. So if I have to reverse this to whatever the DNA was. Oh, okay. Let me, I'm going to cut camera for a second. Maybe that'll help my voice. Someone said I'm cutting out a little. So, all right, again, if I have to reverse transcribe ACU back to DNA, what would that be? So you literally just look at this A, what would be the DNA letter, what would be the DNA letter, what would be the DNA letter that these are opposite of. Let's see what somebody wrote here. TGA, let's see how that sounds. That seems about right to me, right? A T is opposite of this A. And then you have the C here, which means that this must be a G, and then you have a U here, which means that this must be an A. Yeah, and just for the heck of it, ACU, the amino acid that goes with ACU, A, C. Everything in this box with AC is THR, which is threonine. And so now we go back, and I would put Now I might have just screwed that up because, oh no, look, you can still type. All right, you might have to, I would definitely start at the beginning of these so that you don't have to do like more adjustments as this happens. But just be careful, do your best. The one that's really confusing is this one. You're going to have to do a lot of figuring out like how to go from what to where. But really, it's just process of deduction. If you know the amino acid, you can guess one of the RNAs that would stand for it. and then put the DNA that would go with that. There's lots of ones that could stand for leucine if you look at the thing. All right, so why don't you all go ahead and work on that for about 10 minutes, and I can see if people have any questions, and then I'll come back at the end and try to do the DNA part. Hopefully there's time. If not, we might have to save that, because this definitely had to get done today. All right, I'm going to stop the recording.